So this idea, it's very counterintuitive. There was a period of what's known as the OX-Phos Wars. OX-Phos stands for oxidative phosphorylation, and it basically means the mechanism of cell respiration. And there's a lovely quote from Leslie Orgel, who was here in Cambridge in the 1960s. And this actually is Peter Mitchell and Jennifer Moyle, also in Cambridge, 1947 at that. Mitchell had come up with this idea, actually published in 1961, called the chemiosmotic hypothesis. And it's basically this idea that cells are powered by an electrical charge on their membrane. And Mitchell was quite philosophical in his outlook, and he wrote papers that were incomprehensible to virtually anybody. And Jennifer Moyle was a lifelong collaborator with Mitchell, and she was a brilliant experimentalist, a brilliant thinker as well. And I don't think she got the credit that she deserved, so I like to do a call out to Jennifer Moyle. She did all the experiments that made people take Mitchell seriously. And if you read the papers that Mitchell and Moyle published together in the 1960s, in a lot of them in Nature, the ones which were experimental, where Moyle had done the experimental work, I understand every word of it. It's still the terms that we use in our own papers. So it's really Moyle that came up with the nomenclature and the systems and the experiments that are still being used in the labs today. The idea itself, the idea that membranes are charged, was Mitchell's idea. But the fact that anybody takes it at all seriously because the experiments showed it was true is really ascribable to Jennifer Moyle. And what's become clear since then is that the use of electrical charge on membranes to power work, any form of work, is as universally conserved as the genetic code itself. This is something that all cells do. And this quote from Leslie Orgel, not since Darwin has biology come up with an idea as counterintuitive as those of, say, Einstein, Heisenberg, or Schrödinger. These are your mitochondria. They have these amazing membranes inside them, really stunningly beautiful, thin membranes. These are the membranes which have got the charge of the bolt of lightning across all of them. They are just beautiful structures. And here's what's going on inside them. So what we're doing, we're taking out glucose. It doesn't have to be glucose. Food, we keep it generally. We're breaking it down into smaller parts. Don't worry about the names. Spinning it around a thing called the Krebs cycle, which I'm going to tell you a little bit more about. And we're pulling out CO2, which we're breathing out, and hydrogen, not in the form of hydrogen gas, but hydrogen atoms attached to other molecules. But let's keep it simple, but also in a sense more accurate. It's basically hydrogen is being split into electrons, which go as a current of electrons to oxygen, and the protons. So a hydrogen atom is made up of one electron, one proton. The electrons all go to oxygen. The protons go across this membrane, and here we end up with a charge equivalent to a bolt of lightning, because protons are positively charged, and they're all outside. And the current of electrons drives the pumping of protons across there. So that's basically how respiration is working, and that's what's keeping us alive at any moment. You could think of it like a hydroelectric power scheme where the protons are equivalent to the water in the reservoir, where the membrane is equivalent to the dam, and then there's an electrical turbine driving all the work. Now that electrical turbine, talking of revolution, the ATP synthase, it's a molecular motor. It's a rotating motor. It actually does 500 revolutions a second, which is pretty impressive. It's very often found now as a dimer, it turns out, and it bends the membrane. So those fine Christie structures that I was showing, these beautiful extended membranes, they're bent by the ATP synthase itself to be that thin. There's some really beautiful molecular biology of how these systems work being done over the last 10 years. And we now know a tremendous amount about the structure. In fact, John Walker, who's also here in Cambridge, won the Nobel Prize, I think, in 1997 for the structure of the ATP synthase. And Krebs was also in Cambridge for a period. And it was when he was in Sheffield that he finally nailed what's become known as the Krebs cycle, or the tricarboxylic acid cycle, sometimes the citric acid cycle, but I like to call it the Krebs cycle. Krebs was a very charming man, and I do like to remember characters in science. I think it can add a little flavor to what we're talking about. This is the Krebs cycle as I had depicted it right at the end of my book on the Krebs cycle, which I hesitate to say it's on the Krebs cycle, but this is, look at it. I mean, these molecules, these are the carbon atoms, the hydrogens, they're beautiful. What does it mean? Well, what we're pulling out here, CO2, hydrogen, this was really what I was showing you. So there's lots of molecular rearrangements going around this cycle, pulling out hydrogen and CO2, and that's effectively what's going on. I'm showing the molecular structures because this is what Krebs worked out. This was all chemistry. This was the chemistry of these molecules. And so it was hard to do it. Now, those enzymes, I'm just going to mention hydrogen lyase, formic hydrogen lyase, hydrogenase, dehydrogenase. This was Marjorie Stevenson's really big contribution. These things are found in methanogens. I'm going to come on to that in a moment. But this is where she had persuaded Krebs that this is not just one enzyme. This is a whole ecosystem of enzymes going on there. And he goes on to say there are two main results to our joint work. The first is the demonstration that fumarate oxidizes glucose, glycerol, and lactic acid with the same rate as does molecular oxygen. I'm not going to read through to the end of this, but he touches on succinate, he touches on fumarate. Actually, this is about the Krebs cycle. This was the year before he published the Krebs cycle, and this was the work that he was collaborating with Marjorie Stevenson. Now, Marjorie Stevenson had a policy that she never had her name on papers that she had not done direct experimental work on herself. So she is not associated with the Krebs cycle, and frankly, she should be. Because not only was she an inspiration to Krebs, she also was pointing him in the right direction and doing experiments with him at the same time. And she's not remembered for it at all, which is very unfortunate, I would say. Which it may seem, I don't know. I mean, there's always a question about how many authors do you have on papers and who deserves to be on a paper and who doesn't. But sometimes people who have a very noble ambition to only appear on papers where they have done something themselves, then disappear from history in a way which is not fair to them because their contribution was much greater than anybody recognized. So this is Marjorie Stevenson. She wrote a famous book on bacterial metabolism. That's my copy of it. And you may recognize this as the confluence of the great ooze and the Cam, I believe, which is where she was hauling out bacteria in the 1920s and 1930s, archaea, in fact. Paid by the MRC, who wondered what on earth is she up to, dredging the River Cam to get this mud and studying the mud. Well, what was in the mud were the methanogens. And what it turns out the methanogens do, this is not quite right, I'm simplifying it a little bit in case there's anybody in this audience who knows a lot about methanogens, but this is basically it. Look, this should look the same as the last one that I showed you. We've got the Krebs cycle at the heart of it. We've got something that looks like a respiratory chain over here. But no, it's not. This is going in the opposite direction. It's sucking in CO2. It's sucking in hydrogen. It's reacting them together to make the basic building blocks of biochemistry. And it's doing that with hydrogen from the environment. It's sucking in that. But it's also passing it that way as well onto CO2 to make methane as a waste product. And these enzymes that are doing that are the hydrogenases that were discovered by Marjorie Stevenson. So, and that is generating the membrane potential that's driving everything. And from the Krebs cycle, we're making sugars, we're making amino acids, nucleotides. So this is the heart of metabolism. It's not just about making energy, it's also about making the molecules that are needed for life. Now, there are some, I don't want to get involved in much chemistry tonight, but this is what's happening. I've shown you this is what's happening in respiration. We're taking hydrogen from food, we're passing it on to oxygen. Hydrogen's an electron donor, oxygen's an electron acceptor. We end up reducing oxygen to form water. What's happening with methanogens is we've got hydrogen gas, we're passing it on to CO2 to make methane as a waste product. Or we're passing it on to CO2 to make these organic molecules. So this is a waste product generating energy. This is organic molecules. What's happening in photosynthesis? Well, that looks a little bit more complicated, so I'm going to show you an easier version. These processes are exactly the same as each other. We have an electron donor, hydrogen in every case. Electrons coming from hydrogen going to oxygen or going to CO2. So there's a deep conservation of the chemistry, the fundamental chemistry that underlies these processes. They're all basically the same as each other. Once you've got a system which is capable of stripping electrons from somewhere and passing them onto something else and generating a charge on a membrane, that whole thing works. You can go from methanogenesis in a sludge in the River Cam to photosynthesis and everything else. It's a trivial change in chemistry. It's a huge change in outcome. It's a revolution in what happens to the planet, but it's trivial at the level of chemistry. This may intimidate you. Please don't be intimidated. I'm not going to say very much about it, but at the top we have carbon dioxide. This is basically the very heart of biochemistry. And this is the kind of stuff that biochemistry students are force-fed. But all I want you to notice, look, in orangey, browny, sludgy color here, hydrogen, 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 hydrogen. This is, and in green, it's CO2, CO2, CO2. This is the chemistry of hydrogen and CO2. You start with hydrogen and CO2. These are the Krebs cycle intermediates. We've got pyruvate, oxaloacetate. Pyruvate is part of the reverse Krebs cycle. Oxaloacetate, alpha-ketoglutarate, this is part of the Krebs cycle. These are amino acids, which are made directly from it. These are sugars that are made directly from there as well. And these in blue arrows are things that have been done in the lab under prebiotic conditions. This chemistry is spontaneous chemistry that just happens. It's not determined by genes. It's not controlled by enzymes. This is chemistry that can be made to happen with metal ions as catalysts, and that's more or less it. Not the entire network all in one go, but pretty much everything in the blue arrow has been done in the lab as that one step. So the chemistry of life, I've got this on the right-hand side, if you can see it, the chemistry of life is actually older than the genes that are supposed to encode the chemistry of life. That's a really interesting thing. And the genes in archaea and the genes in bacteria, they're very often completely different, but they're catalyzing the same chemistry. This chemistry, these reaction steps are different genes in bacteria and archaea, but they're catalyzing the same underlying chemistry. That chemistry is older than the genes themselves, and it's spontaneous, and it will happen in certain environments. Another thing I want to tell you before I touch on the origin of life a little while is that coming out of this spontaneous metabolism, there are also patterns in the genetic code which suggest that there were direct interactions between amino acids that were popping up in this metabolism and the nucleotides that were also, hopefully, popping up in this metabolism, interacting directly together, which means if you've got a random sequence of RNA, for example, you'd have a non-random sequence of amino acids that might bind to it, or they might bind into a pocket, we don't know exactly. But if you were to zip all those up together to form a polymer, then this random sequence would would template a non-random peptide. If that non-random peptide could do something, act as an enzyme, a catalyst, then it can be selected for. So just thinking about these basic metabolism before we get to genes, before we get to information in biology, we have this metabolism giving rise to the building blocks for all of this, and this is beginning to be where information is coming in. Let me frame this in terms of the origin of life itself. What I'm kind of getting at here is that electrical charge on membranes, electricity, let's say, drives metabolism. We don't need genes for this metabolism. It's driving it anyway, which gives rise to genes. So the information is coming a little bit later. And for that to be true, it has to all happen in one place. There has to be a place where you can have electrical charges driving metabolism, giving rise to genes. That really, well, that sounds preposterous, doesn't it? But it's really not.